Let me begin with a situation that almost every peptide development team encounters sooner or later. You've invested heavily in peptide synthesis, optimization, and early studies, and everything looks promising. But the moment you prepare for an FDA or Health Canada submission, a new question appears on the table. Why is peptide characterization so expensive? And how do we plan for it correctly? This presentation is designed to answer that exact question. We'll explore what actually drives the cost of peptide characterization, how regulators think about your data, and how experienced teams budget smartly without compromising compliance or timelines. To understand cost, we must first understand regulatory expectations. Both the FDA and Health Canada place extremely high importance on peptide characterization because peptides are inherently complex and sensitive molecules. Regulators want complete confidence that your peptide is correct in identity, consistent in purity, biologically active, and stable throughout its life cycle. Although FDA and Health Canada largely align with ICH guidelines, the FDA follows a fit-for-purpose approach. This means the deeper the complexity of your peptide, the deeper the analytical justification required, and that directly influences cost. One of the most underestimated cost drivers is peptide complexity. Simple linear peptides are comparatively easier to characterize. But as peptide length increases, or when you introduce modifications, conjugation, or structural folding, analytical complexity rises sharply. Complex peptides often require peptide mapping, multiple mass spectrometry workflows, advanced data interpretation. From a regulatory perspective, complexity means higher risk, and higher risk demands stronger analytical evidence. That additional depth is what drives time, effort, and cost. Every analytical technique carries its own cost structure, driven by instrument time, expertise, and regulatory documentation. Regulators rarely accept conclusions based on a single technique. Instead, they expect orthogonal confirmation, meaning different methods validating the same attribute. That's why many submissions rely on combined analysis packages, where multiple techniques are strategically selected and bundled together. Choosing the right combination of methods, not simply more methods, is where experienced analytical labs add real value. Across FDA and Health Canada submissions, 6 factors consistently influence cost. Peptide complexity, choice of analytical techniques, regulatory compliance requirements such as GLP or GMP documentation, sample handling needs, especially for lyophilized or unstable peptides. Turnaround time. Rush timelines always cost more. And finally, the level of documentation required for submission readiness. Teams that understand these factors up front gain much better control over both timelines and budgets. Successful organizations treat peptide characterization as a planned regulatory strategy, not a last-minute checkbox. They define regulatory needs early. They assess peptide complexity honestly. They consult experienced analytical labs during development, not after problems arise. They plan for contingencies, and they always request detailed, itemized quotes. This approach minimizes rework, prevents delays, and protects project timelines.
Cost optimization is not about cutting corners, it's about planning intelligently. Bundling analyses, avoiding expedited timelines, and ensuring proper sample preparation can reduce overall costs by up to 30%. At the same time, teams must be aware of hidden costs, such as reanalysis fees, method development, rush charges, and additional regulatory documentation. A modest budget buffer early on often saves months of delay later. When planning peptide characterization, many teams budget only for the planned tests, but regulatory reality often adds more. Hidden costs usually come from reanalysis if results are unclear, rush charges when timelines tighten, or method development for complex or modified peptides. Additional documentation may also be required when regulators ask for deeper data interpretation during review. The key lesson from real submissions is simple. Always plan a contingency buffer. A small buffer protects timelines, avoids last-minute stress, and allows teams to respond confidently to regulatory questions without disrupting the project budget. Peptide characterization is not just about running tests, it's about generating data regulators trust. Resolve Mass Laboratories combines advanced analytical platforms such as HPLC, LC-MS, MS, and MALDI-TOF, with strong regulatory expertise aligned to FDA, Health Canada, and ICH expectations. All analyses are designed to be submission-ready, supported by validated methods and GLP/GMP documentation. With transparent pricing, flexible scope-based quotes, and no hidden fees, ResolveMass acts not just as a testing lab, but as a regulatory partner supporting efficient, confident submissions. To summarize, peptide characterization costs are driven by complexity, regulatory expectations, and analytical depth. Early planning, transparent budgeting, and collaboration with experienced laboratories turn characterization from a regulatory hurdle into a strategic advantage. When done correctly, peptide characterization not only supports approval, but accelerates it. Thank you for your time, and we look forward to supporting your next FDA or Health Canada submission.